Hey guys, this is Mr. O'Brien. This is going to be a Molecules of Life review. So this kind of covers chapter 3.2. Now, molecules of life are kind of important, and a lot of kids struggle with this. But when we talk about molecules of life, we're talking about important macromolecules inside your body that help you do different things. So we're talking about carbohydrates, proteins, lipids, and nucleic acids. So let's start here with talking about carbohydrates. Now, carbohydrates are simple sugars, and what this means is when you eat food, when you eat anything that is sugar, we're talking about carbohydrates. So what makes a carbohydrate is a sugar makes a ring structure. Okay, glucose, fructose, sucrose, all these things are forming a ring. But what really makes it easy to identify is that carbohydrates form this 1 to 2 to 1 ratio. And what that means, for every one carbon, there's 2 hydrogens and 1 oxygen. So when you hear this 1 to 2 to 1 ratio, that is what they're talking about. So the one molecule we do want to talk about is glucose. So glucose, very common, especially in plants. Animals can use it as well. But its chemical formula is C6H12O6. Now, when we break this down, we look at these 6, 12, 6. Each one of those you can divide by 6, obviously, and this is where you get that 1 to 2 to 1 ratio. So glucose looks something like this. Now, we talked about it forming these ring structures. So it's a carbon ring structure, and there's some oxygens that are kind of in there as well. But the carbons and oxygens kind of form this nice little ring. And this, it's obviously simple there. Obviously, it's not a perfect circle, but it does form a ring in that structure. Now, off of some of those bonds, again, we have, hydrogens, OH groups. Same thing here, hydrogen, OH group. Same thing here, hydrogen, OH group. But off one of these carbons, we have our functional group. And in this case, we have a CH2 and an OH. Now, this functional group right here is what gives it its characteristics. So all sugars are a lot of sugars are C6H12O6. We call those isomers. Same structure, or same formula, but different structure. But that functional group right there. Now, again, we're looking at rings right here, and that's really what we want to, the take-home message we want to bring home here. So carbohydrates, its monomer or single unit we have are what are known as monosaccharides. So the always way I remember it is mono means one, saccharide means sugar. Now, Mr. Peanut here. Okay, we all know who Mr. Peanut is. He wears a nice little top hat, and what does he wear? He also wears a monocle. And mono meaning that one lens that we have right there. When we have two joined together, we call that a disaccharide. Okay, two. And when we have multiple of these things joined together, we call it a polysaccharide. So sugars are kind of easy here. Okay, they all have the same sugar base that we want to look at. So if we break this down, monomer, one single unit that we have right there. Disaccharide is when we have two of these bonded together. Polysaccharide is when we have a nice long chain of sugars bonded together there. So that's kind of the difference that we want to see here. Now, what carbohydrates do is they provide quick energy. All right? And carbohydrates, if you want to think about it, like sugars, you eat a candy bar, it provides you that quick burst. You feel that energy, you feel that buzz in the back of your skull. Now, a lot of weightlifters, they do things called pre-workouts. And pre-workouts, it gets you ready for that workout. Again, this is a high sugar drink to kind of get your muscles that quick boost of energy to get a good hard workout in. But you got to remember, quick energy, all right? So sugars, you're using that, you're metabolizing it quickly to do certain tasks. Now, proteins are in food like red meat, fish, chicken, all that sort of stuff. And proteins are really used to build support, build muscle, build molecules, but also to provide signals for your body. So proteins are made of carbon, hydrogen, and oxygen, just like carbohydrates, but the difference is nitrogen. So there's nitrogen in it. Now, this is a picture of a protein right there. It kind of folds very differently, and depending how it folds depends on how your body sees it. Now, again, they're used for organization, they're used for movements, and proteins are actually what enzymes are made of, and those are used to catalyze reactions or speed up reactions inside your body. Now, our monomer of proteins is what's called an amino acid. So the single unit that we have in proteins is an amino acid. When we have a long chains of amino acids, we call them polypeptides. And polypeptides are just a long chain here. So again, same type of structure. Amino acids 1, polypeptides is when we have a long chain of them. Okay? Now, polypeptide bonds are how these amino acids form together. And we can kind of see this down here. And what we're doing is we're taking that water molecule out of these two things. Now, if you remember from last section, this is called a condensation reaction. And in this reaction, we're taking out water, as we see right here, and we're forming that bond, as you can see down here. So, those bonds are forming, and that's how we create this very long chain, using that peptide bond. Okay? So our amino acid, it's based off a carbon. So it's one carbon with 4 different branches coming off. So the one branch we have is a simple hydrogen. Really doesn't do anything, just kind of a place filler. Now over here, we have what's known as our carboxyl group. Now in the carboxyl group, all right, we have a carbon, double bond, an oxygen, and an OH. Now this group right here, okay, the way I always remember it is carboxyl, okay, has the word pretty much carbon in it. So carbon right here. All right. Now the important one that pretty much gives the amino acid its characteristic is something called the functional group. And I'm just going to draw this as an R. And the reason I'm drawing it like that is the functional group changes depending on what amino acid you have. Okay? So this functional group, or these amino acids, there can be a total of 20 different amino acids. Okay? Now, this functional group changes, and depending on what it is, depends on which one of those 20 amino acids we have. Now, the last one we have is what's known as the amine group. And the amino group is a nitrogen bonded to 2 different hydrogens. Now, when I look at these, I kind of got to know carboxyl functional amino acid, or amino group, excuse me. Now, amino has an N in it, just like there's a nitrogen in there. Carboxyl has a carbon, carboxyl group, and the functional group is the one that's changing. So this is your basic structure of all amino acids that we have. Now, proteins, like I said, make up enzymes. And these enzymes, sometimes they're called biological catalysts. But let's look at an example here, and this is called a lock and key model. Okay? So here's my enzyme, and what happens is we have a molecule called a substrate that comes in that takes the same shape of it. Okay? So we're going to call this our substrate. This is, what's coming in to form whatever we are doing in that chemical reaction. Now, the place where that reaction takes place is called the activation site. So this is where, this either, splits in half, to form two different things, or maybe two different substrates are coming in to form one, but it happens at that activation site. And why this is important is it speeds up the chemical reactions, and it's not consumed, so it's able to be used over and over again. Now, lipids, there's 5 different lipids that we want to look at, and lipids are fats. So, went too far. Now, come on. So our lipids are nonpolar. And why this is important, and really, why I'm even talking about it, is because that means they're not able to dissolve in water. So water is a polar molecule, which means it has opposite charges. So lipids are nonpolar. This is why when you put, you got a glass of water and you put oil into it, what happens? The oil floats at the top. Okay, so it doesn't mix, it doesn't want to associate with each other because of that nonpolar structure. Now, lipids are made of carbon, hydrogen, oxygen, and phosphorus. So the difference here is that lipids are used for long-term energy storage. And if you think about it, right, what happens when you get, like, fat, right? So you get a belly, right? What is that belly? That belly is the accumulation of lipids. And why is that there? You ate too many calories. Those calories are now being stored in case you need long-term energy. Okay, so once you use all your carbohydrates, that's when you shift to using the lipids. All right. Now, we have 2 types of fatty acids. Now, this is the first type of lipid that we have. Now we have saturated and unsaturated. And let's look at this picture here to kind of describe it. So I always tell the kids to look at this one of the carbons, and you'll notice how many bonds does this carbon have. It could form 1, 2, 3, 4 bonds. So it's attached to 4 different chemicals. That's what's referred to as saturated, where that carbon can form 4 bonds very easily. Now unsaturated, you'll see this can only form 3 bonds. So that's usually by either a double or a triple bond, and that's what we talk about unsaturated. So saturated is all single bonds, and unsaturated is double or triple bonds. Okay, so that's really the only difference. Single bonds. This is what we call a saturated carbon. Can form 1, 2, 3, 4 bonds. These carbons right here, if I could draw, these carbons right here are what are known as unsaturated, not having its complete 4. So fatty acids is a very long chain of carbon. So triglycerides, a little bit simpler. Well, it's kind of a branch there that we have here. Triglycerides, we have this glycerol molecule, and what's attached to it is 1, 2, 3 fatty acid chains. So if you see 3 tails, you got to think of triglyceride. Now, just like a tricycle has 3 wheels, triglyceride, 3 chains attached to a glyceride. So phospholipids, a little bit more important. This is what's found inside of our cell membrane. But we have this phosphate head. We have these phospholipid tails. Okay, these are our lipid tails. Now, the head is polar, which means it likes to be associated with water, while our tails are nonpolar. So we form this type of structure where the heads are associated on the inside and the outside of the cell, whereas the tails kind of work in the middle. And what this does is it gives it this nice structure for what's known as our phospholipid bilayer. Okay, so we have 2 fatty acid tails, 1, 2, attached to that glycerol molecule right here. Okay, now again, this is what's found in our cell membrane. This is kind of why it's important, why we want to talk about it. So phospholipid, we have a head, we have 2 tails. These are nonpolar, and our tails, or our heads, excuse me, are polar. So they are the ones that are associated with it. Okay, and again, we have the hydrophobic tails. Okay, hydrophilic heads. Okay, so that's just, if we want to look at this, that's what I kind of talked about a little bit earlier there. So waxes are kind of a protective substance used by animals and plants. Now they're waterproof, just like in a beehive. If you ever use beeswax, it's a waterproof substance. But different plants, different animals, they use this as a protective layer to keep water out. And waxes, they have this alcohol chain right here as well as a fatty acid chain, and that's kind of the structure that we see with waxes. But the key here is they're waterproof substances, and they perform those, or they form those protective layers inside of cells and out of cells. Now steroids are another one that are really easy to identify, and the reason for that is because steroids have 4 carbon rings that are attached. You'll notice here 1, 2, 3, 4. Okay, this is cortisol. Now, a lot of people get cortisone shots to make themselves feel better, and this is the structure. Now, you'll notice each one of these is a little bit different, but they all have these 4 chains. Now, cholesterol is another one that is a steroid. 1, 2, 3, 4, 4 carbon rings attached to each other, and that's kind of its distinguishing factor. Okay. Now, our last one we want to talk about is nucleic acids. Now, nucleic acids, why they're important is it allows us to store information in our body. Like our DNA stores all of our genetic information that makes us who we are. So, the monomer of nucleic acid are what are known as nucleotides. Okay, and nucleotides are that single substance of these. So if you notice in DNA, we have all these different colors. Each one of these, let me get a color I can draw. Each one of these is its own monomer, and those are called nucleotides. Now, if we want to draw this, right, nucleic acid, each nucleic acid is composed of 3 parts. And the first one is we have a sugar molecule. Okay, and that sugar molecule is a carbon ring, just like all sugars. Now, attached to that sugar, we have our nitrogenous base, and attached to the back end of it, we have a phosphate. Okay, this is called our phosphate group. So, composed of 3 parts, we have phosphate, sugar, nitrogenous base, and together all these form our information inside of our body. So, I hope this little tutorial helps. It kind of gives you a little bit more of an explanation for each of those molecules of life. So, this is Mr. O'Brien signing off. Good luck.